Our last presentation. I've been waiting all day to learn more about cockroaches. So I know I won't be disappointed. I'm very excited. Mauri's also a former German student, so I'm really been looking forward to this. Mauri Padilla is our next presenter, and he worked with Professor Daniel Gold on a research project titled, "Is the Madagascar hissing cockroach infecting gangrene? Oh, Barbicola migrator, susceptible to the antimicrobial drug paramomycin." We're gonna stick with that. So Mauricio sent in a video of, on his topic, and so we'll watch that before I turn things over to Professor Gold and Mauri himself for a question and answer session.
Hello, everybody. Thank you for coming to my presentation today. My name is Mauricio Padilla, and I'd like to talk to you today about my work on the Gregarian parasite Barbicola migrator and its host, the Madagascar hissing cockroach. So I'd like to start by talking about the infectious and parasitic disease category of the global health burden and how it accounts for such a large portion of the global health burden. So in this category, there are a number of fairly severe and fatal diseases that are attributable specifically to apicomplexans. So what are the apicomplexa? Well, essentially, they're a large phylum of eukaryotic parasitic protists that are unicellular, so single-celled. Now, this is a very diverse and abundant group of organisms, so diverse and abundant, in fact, that there are estimates out there that postulate that all animals on Earth are host to at least one apicomplexan. So another way to highlight just how prolific and widespread these organisms are, a research group did an ecological study where they tried to determine a diversity of an area by searching for organismal DNA in soil samples. And what they found is that over 50% of the organismal DNA in these soil samples was attributable to apicomplexans in general. So I'd like to talk a little bit about some of the infamous apicomplexans that you might have already heard about or known about in some way. So you have toxoplasma, which causes toxoplasmosis. This is a parasite that infects cats and humans and is part of the reason why doctors advise pregnant mothers to stay away from cats and cat litter if they can. So another parasite is known as Plasmodium, and this is a parasite that is carried by mosquitoes and actually causes malaria. So this is a very well-known, very well-characterized parasite. So these two cause very severe and fatal diseases, and much is known about their lifestyles and how they parasitize organisms within their cells. So they live what's called an intracellular lifestyle, where they invade a particular cell and then parasitize it from within. I'd like to talk now about Cryptosporidium and focus a little bit on this branch of the apicomplexa. So Cryptosporidium causes cryptosporidiosis, and it infects vertebrates. What's different about Cryptosporidium from some of the other branches is that recent phylogenetic work has shown that they're actually very closely associated to Gregarians. And our lab actually focuses our studies on Gregarians. But what's important here is that these two groups have a epicellular lifestyle. So instead of parasitizing their hosts by invading their cells, they'll attach themselves to the lining of a tissue and then without invading the cell, obtain nutrients that way. And there's not a lot known about the mechanisms behind the way that they attach themselves and develop. So these aren't as well characterized. But what's important to know is that Cryptosporidium causes cryptosporidiosis, which is a diarrheal disease. Now, diarrheal diseases make up almost a third of a portion of the infectious and parasitic disease category. So it's important to understand how epicellular parasitism works. So luckily for us, Gregarians are very similar to crypto, except that they infect invertebrates. So other than that, they have a very similar epicellular lifestyle. They have very similar oral fecal transmission systems, so they infect in much of the same way. And they have very similar lifestyle, life stages and development. So now I'll talk a little bit about what the Gregarians are and a little bit about them in general. So the Gregarians are the most diverse group of apicomplexans. So diverse, in fact, that they infect just about every invertebrate you can think of, except for nematodes, which are little microscopic worms, and tardigrades, which are those radiation water bears that you might have seen on Planet Earth or BBC. But essentially, Gregarians affect every invertebrate you can think of, from terrestrial to aerial to aquatic. And they range in variety and shapes and sizes, but our specific Gregarian infects the intestinal lining of its host. And as you can see here on the picture on the right, this is a picture we took from our lab, where you can see just how massive the Gregarians are. So Gregarians, like I mentioned earlier, are apicomplexans, so they're single-celled, and one Gregarian can be as large as 1 millimeter in length, which is massive for a single-celled organism. So now I'd like to explain why we want to use Gregarians as our model organism to understand epicellular parasitism, as opposed to just studying cryptosporidians. There's a few reasons for this. One of the biggest is the financial strain that using cryptosporidians and infect invertebrates has. So it's way cheaper to study Gregarians, which infect only invertebrates, because it's a lot less expensive to maintain a breeding colony of cockroaches than, say, a breeding colony of laboratory mice. Another big reason is it reduces the risk of infectivity to the researcher. So because Gregarians only infect invertebrates, you eliminate the risk of the researcher becoming infected and developing the diseases that might be associated with epicellular parasitism. So now I'll talk about the reasons why we chose specifically Bulbaricola migrator and the cockroach host as our designated model organisms for research. And there's two parts to this, essentially. The first being the Madagascar hissing cockroach is only infected by one type of Gregarian, which is the Bulbaricola migrator. So this is important for future experiments later on, where we need to know exactly what species, and only know if one species has a certain function in experiments to be designed later. Another important reason is because the cockroach host is a fairly large invertebrate and is very durable and sturdy. This is important because some of our plans and directions that the lab is heading towards involve a number of surgery techniques that we'll use to try to directly implant genetically modified parasites into the cockroach. So we believe that the cockroach is a better invertebrate for withstanding and surviving those kind of surgical procedures. So now I'll talk a little bit about the life cycle of our parasite, the Bulbaricola migrator. So it's very simple, and I'll start at the very beginning with like the egg stage of the parasite. So essentially, inside the cockroach, you have these growing parasites, and they get excreted in an egg-like casing called the gametocyst, and they'll be excreted with the dung of the cockroach into the environment. Now, these gametocysts are very environmentally durable, as they should be if they're going to survive the extreme environment that they're usually located in the Madagascar area. So from the gametocyst stage, they'll undergo a process known as dehiscence, where pores inside of the gametocyst will open up and allow for these strands to shoot out in every which way into the environment. Now, these strands contain the infective stage of the parasite, known as the oocyst. Now, the oocyst is linked up in these strands by the hundreds and thousands as singular beads in a chain, and each oocyst contains inside of it 8 individual parasites in the sporozoite stage. So I'll try to go through this slow so we get a clear understanding. So anytime a cockroach eats an oocyst, they're eating essentially 8 parasites. So what happens is the oocyst will land on some of the cockroach food, and the cockroach will inject it, and then these microscopic oocysts will go through their digestive system, and once they reach about the mid-gut, there's something inside of the cockroach intestinal bile that triggers the next process of development, known as exustation. So during exustation, the oocyst is opened up, and the 8 sporozoites inside, the 8 parasites, they look like tiny little worms. they'll travel along the intestine of the cockroach, and then they'll attach themselves to the intestinal wall, the epithelial cells. Once attached, they'll obtain their nutrients and mature into fully grown trophozoites. And then once they're in the trophozoite stage, they'll undergo their form of reproduction called syzygy to start the cycle all over again. So I mentioned earlier that the main goal of this lab is to gain understanding of epicellular parasitology and to use Gregorians as a model for that methodology. So we would like to replicate some of the work that's already been done on Cryptosporidium in understanding epicellular parasitism. So there's a lab that has established a tool that helps them understand specific genes of interest, and they use what's called a transgenic expression system. The idea behind it is that they have a circular piece of DNA, and inside of that circular piece of DNA, they're able to add in a gene called neomycin resistance, and this is important in a second. So they'll use a process called electroporation to insert this gene inside of the sporozoite stage of the parasite. They'll then directly implant the parasites into their rat hosts, and then they'll dose the rats with a drug known as perimomycin. Now, perimomycin is used as their selectable marker because it inhibits and prevents the growth of the sporozoites into the oocyst stage. So effectively killing or inhibiting the growth and further reproduction of the parasites. However, parasites that are genetically modified have this neomycin resistance gene. This neomycin resistance gene confers resistance to the drug perimomycin. So essentially, when the rat excretes out any oocysts, the idea is that these oocysts will be genetically modified because they survive the dosing of the perimomycin. So the lab has been able to continue this cycle through a few more stages, both growing the oocysts in medium and then reinfecting them to get purer and purer genetically modified parasite samples. We would like to replicate a similar kind of transgenic expression system to study Gregorian models. So my part in establishing this transgenic expression system relies on developing the selectable markers. So I essentially try to determine how we can select for the genetically modified parasites. Now, the easiest way for me to do this was determining if perimomycin is as effective for Gregorians as it is for the Cryptosporidians. So in my initial experimental design, the idea was that I would take 4 groups of cockroaches of about 15 each, or 30 each to reach statistical significance, and then split 2 of those groups into an infected category, and then 2 of those groups into an uninfected category, where then I would treat 1 group in each category with perimomycin and the other with no treatment, essentially giving them the same solution but without the perimomycin drug. I was then to quantitate how many oocysts they excreted in their dung, as well as at the end of treatment, dissect the cockroaches and count up how many internal stages, trophozoites, were present in each cockroach. So when we started this venture, we actually didn't have the cockroach numbers to really sacrifice over 60 cockroaches that we needed to for the experiment. So we wanted to see if there was a general trend in perimomycin so that when we had upscale of our cockroach numbers, we could do this for real with a better understanding of what kind of dosage to use. So in my experiment, I took 2 groups of cockroaches of 15 each, and I dosed one with perimomycin in an 8% sucrose solution, and I dosed the other one with just an 8% sucrose solution. And then I only quantitated the internal trophozoite stages to see if there's some kind of general effect on the trophozoite stages of the parasite. So after collecting the data on the samples, this is the data I collected, and what we noticed were 2 things. At first glance, it seemed that we definitely had a smaller average in parasite count in the treated population than we did in the untreated, but we noticed that we lost about 1/3 of the sample size during treatment for the perimomycin treated group, and we only lost 1 sample in the untreated sample group. And then we had the spread of data in both groups that were similar that we couldn't really account for. So we came to 2 conclusions. One was that we needed a better way to select a population that wasn't as old and as big, that would help reduce the number of cockroaches that died during the experimentation. And two, the way to account for this data is we needed to ensure that the cockroaches were equally getting the amount of permamycin dosage as every other cockroach. So I conducted a consumption assay where I measured how much solution each cockroach would drink in about 24 hours. And what I found is that they were very similar in that they both drank roughly 220 microliters, but in finding that they roughly drank about 220 microliters a day, we determined that delivering permamycin through water solution was probably an inefficient method of delivery. So this past semester, before schools closed out, I was in the works of developing a protocol to manually inject the permamycin into the intestine with a syringe. So the idea would have been to equally administer permamycin to every cockroach in this way and effectively determine whether or not permamycin actually had inhibitory effect on the parasite. So now I'd like to talk a little bit about some of the projects I've been involved with other than my own, and Caitlin Stark's project, where she's been leading a microscopy project and talking about discovering the cellular structure of the parasite and a little insight about how they interact with the hosts. And as I'd like to mention Bellini Han, Gideon Hale as well in their existation project, and how they essentially are another very pivotal part of the transgenic expression system. Their project involves trying to efficiently optimize the way to gain a sample of sporozoites so that we can genetically modify a large group of samples at a given time. I'd like to thank Dr. Gold and the Natural Science Foundation, as well as the Brother Romard Scholarship donor, for helping fund my research. And I'd like to highlight Dr. Richard Clofton and Dr. Tamara Cook for helping me to develop some of the methods and protocols to generating and maintaining the Peregrine parasite colonies. I'd like to answer any questions you might have now. Thank you.
Mauri, thank you so much. I have a bunch of questions. I imagine our audience members do too, but before we toss questions your way, I wanted to give Professor Gold a chance to say a few things about working on this project with you.
Can you guys hear me? So I'm both extremely happy and extremely sad. So Mauri has been with me since I basically started this project, or starting the lab. I think he was essentially one of the very first people to work with me when all of this was sort of a figment of a dream. We didn't have parasites, we didn't have roaches. In fact, we tried doing this in beetles first. We couldn't find parasites. We had no methods. We didn't know how to find them. We didn't know what to, I mean, his work went so far beyond just the project that he's talking about here, which he was doing for his honors thesis. He went in essentially, and I told everyone who worked with me at the time, I mean, I can give my best advice, but I've never worked with these before and I don't know how to do it. And so we're going to learn together. And that's a scary thing, especially in science and especially in biology. And so all of my students, especially Mauri and Caitlin and all the ones who, were there from very early on, played a big role in just sort of developing how we do things. And the fact that we got to the point that we are is in no small feat to Mauri and Caitlin and other honor students in the past, like Nick Barrett and Matt Fensel. And so I just want to give that context. And when he said at the end of those other projects, Mauri had a really big hand in either co-developing or having crucial assistance to those other projects. He's been sort of somebody who I've relied on to sort of help, mentor other students, as well as work on his own project. And he's just a pleasure. I mean, Mauri's just a pleasure to work with. And again, it's not the typical situation where I have all this knowledge that do this, this, and this, and this, and this. It's very much, was something that he had a big role in building from the ground up. And I'm very appreciative and, again, very sad, but also very happy because, he's done such a great job and he's, been wonderful. That's what I have to say.
Thank you so much, and thanks for working with so many of our students in your lab and supporting them. Mauri, you have a number of comments coming in about how well you're doing. If anyone has questions for Mauri, if you could enter them in the chat, that would be great. And Mauri, I want to add how impressive that was as an outsider. I often think about science researchers trying to answer a small portion of the questions that you presented of sort of does paramomycin have the effect X or Y? And I didn't realize going into your project how much work you had done to really develop completely new methodologies. It's really, really impressive. And we have our first question coming in from Professor Hammonds, who asked, what was the toughest part of your research? Hang on, Mauri, I think I need to unmute you. Okay.
Hi, so thank you everyone for coming. I appreciate you being here. I know it's kind of later in the day. But Professor Hammond, as far as the toughest part of the research, I'm not sure I have one specific answer. What's kind of fun is that there is always something, one problem or hurdle or something that we had to kind of learn to work around. So initially, as Dr. Gold mentioned, the project started on beetles. And so our problem there was that we couldn't find any parasites. But if you're talking about my specific project, probably the most frustrating part and the part I'm a little sad that I didn't get to finish was developing that methodology to accurately inject the paramomycin drug into the cockroaches. I was having some problems before spring break trying to find a way to really do that accurately and precisely for a given number of roaches. And I was planning on kind of perfecting that methodology over spring break, and it just wasn't able to happen. But yeah.
Mauri, this is a kind of a silly question, but I'm curious because you said that the one advantage is that the cockroaches are more cost effective than working with mice, for example. But you also said that one of the challenges was that you didn't have an unlimited supply of the right kind of cockroach. So can you tell us, like, how hard is it to get the appropriate type of cockroach, or how much does it cost to get sort of science grade cockroaches?
I feel like Dr. Gold could explain a little bit more about maybe the finances about that. But as a worker in the lab, part of the problem we had with cockroaches specifically was that they actually live about 2 to 3 years, and then they reproduce fairly frequently under the right conditions, which we ended up finding and had an overabundance almost. The other issue is that the time it takes for them to develop into maturity is about 7 months. So when we initially had our colony, we only had about 100 or so cockroaches. And while they were reproducing, it was rather slowly. And then we didn't really have the time to wait 7 months for their offspring to reach maturity. So at that point in time, I didn't really have the numbers to be sacrificing over half of the population for only my project.
Thank you. That's all fascinating. And you have one more question from another Mauri Padilla, I think is your dad, the best kind of audience member to have, who's asking you, moving forward, what's the best advice you would have for team members who are going to take over this project?
So I would advise to kind of don't take for granted some of the work that's already been laid out. Caitlin Stark was very crucial in helping me over the summer, specifically with finding the internal stages of the parasite. So we were able to expand into a whole number of projects solely because of that discovery. All the work that I did with Nick Barrett and Matt Fensel in the past really helped me kind of get into a routine and consistency here. So I hope that moving forward, the next group of Dr. Gold's lab can use that and then can develop their own methodologies to pass on further along.
Dr. Gold, thank you so much again for supporting Mauri, and Mari, thank you so much for sharing this with us. Congratulations on completing your thesis, completing the honors program. This is usually when Dr. Gold would get to give you your honors stole. And we really regret that you can't, he's doing it on the screen. It's in the mail, so if you don't have it already, you should get it in the next couple of days, but congratulations.
Thank you.
And thank you all so much for being here. This has been a wonderful symposium. It's a shame that we can't be together in person, but I'm so grateful for everyone who came on Zoom, and the students and advisors who put in their work to make sure we could have this virtually. It's really been a highlight of a very weird semester. But thank you so much.